Okay, so I think the recording is on at the moment. So first of all, welcome everyone to this new seminar series, the first one today of the new year, 2023. And I would like to start presenting the speaker of today. So I will try sharing again my screen. I think you can see my slide, right? Can you? Yeah?
Yes, we can.
Okay, thanks. So today we have 3 speakers that are coming from the lab of Sara Mitri in the University of Lausanne, Aurore Picot, Oliver Mikok, and Sara Mitri actually, and they will talk about how do microbial species interact and how do they find out. So something regarding the lab and the interest of Sara Mitri specifically. So she works on computational sciences and biological evolution. She also worked on applied evolutionary thinking to the designing of computational algorithm. And generally, she's interested in ecology, evolution of simple microbial communities, and how to reveal the logic of how microbial ecosystems function. So she had a PhD in computer and communication sciences, then a postdoc at Harvard and Oxford. And then she became independent research fellow at the University of Lausanne, where also she got a tenure track assistant professor, and I think she's actually sitting now there in University of Lausanne in the Department of Fundamental Microbiology. So that's everything for the introduction, and I will stop sharing my screen, giving you the ability to share yours, and please go on with your talk. Thank you for being here, and please, stage is yours.
Great, thank you very much, Marco. Thanks for the introduction and obviously for the invitation. It's a very nice seminar series, so it's really great to be part of it. And also I thought today we could do things a little bit differently than usual. So since it's all virtual, I figured it would be nice actually to bring in Oli and Aurore. Instead of talking about their work myself, I would let them introduce it, since they will be much better at explaining their own work than I can ever be. So I will go ahead and start then. So I'd like to start sort of thinking about questions quite generally in ecology, since I suppose a lot of the people in this audience will be thinking about other ecosystems. So in ecology, we might think about natural and man-made ecosystems, and we might ask ourselves whether the dynamics that take place in these ecosystems, whether they're predictable. And whether we're able to predict these kind of dynamics relies on previous work that's been done in ecological theory and experiments, of course, and whether we've been able to discover universal laws of ecology. And of course, we have quite a few theories around that help us to make these kinds of predictions, but probably we would be lacking the resolution to be able to make quantitative predictions and really know if things like climate change occur, which species would go extinct, and what would the state of the ecosystem look like in the future. And so this sort of motivates the question whether one can actually predict things in a way that we would be able to control them towards a desired outcome. So for example, to maintain certain species diversity in an ecosystem, or whether we would be able to design new ecosystems from scratch. And so in my lab, we're interested in all of these questions, but not in these larger ecosystems, but rather in microbial ecosystems. And that's partly because these are easier to handle, because they occur over relatively short timescales. And then also because they have a lot of relevance. So for example, we know that in the gut, so this is a picture of a honeybee gut, we know that the microbes provide nutrients to their hosts. We know that microbes in the soil can help the plants to grow, and they're also, of course, involved in producing our food. So there's also an important relevance of understanding microbial ecosystems. But rather than studying these complex microbial ecosystems, in my group, we prefer to keep things a little bit simpler so that we can actually get a handle on what's going on and measure how the population dynamics of all the constituent members. And so we work with synthetic microbial communities, where we take a few representative isolates of a given ecosystem and then culture them in simple experiments in the lab and see how they affect each other. And these synthetic communities have actually become quite popular. So if you look across systems, you will find that there have been many such synthetic communities that have been constructed. For example, like for the honeybee gut that I showed you on the slide before. And the one down here is the synthetic community that we work on, and I'll explain that in a little bit. But first, going back to the question, so why is it that predicting ecosystem dynamics, in particular in microbial communities, why is it so difficult? So one of the issues is that we don't actually know, if we throw a bunch of species together in the lab that we've isolated from the environment, which ones will tend to coexist, and can we, what do we base these expectations on? And a sort of obvious place to start thinking about this is thinking about their interactions. But we don't actually understand enough about how microbes interact. And if you ask a given person in the field, will two microbial species tend to compete or cooperate, there will be very different answers depending on who you ask. So this is sort of one of the first questions that we need to look at, is how microbial species interact, and how do we actually go about measuring them and quantifying them? Because again, depending on who you ask, they'll come up with different answers, because the way we measure things are different. So this will be the focus of the talk today. So first, let's define what we mean by interactions. And here, what we're talking about is the effect of one species on the growth and survival of another. So if we think of sort of a familiar predator-prey system, then the effect of the predator on the prey is negative, and vice versa is positive. So this is sort of a simple thing to keep in mind if you lose track. And of course, a lot of the thinking in theoretical ecology has been based on these microbial ecosystems. So we tend to think of things in sort of these trophic levels, where you have sort of predators that compete amongst each other, but then have plus minus interactions with the next trophic level. So their prey, and then this can continue to the next trophic level down. Now, when we started looking at microbes, perhaps 10 or 20 years ago, then we saw that there seems to not be as much of a clean division between trophic levels as we would see in macroecology, but instead the relationships seem to be a little bit more complicated, and you cannot sort of categorize microbes into these levels. So for example, you might have one species that provides food for another, but poisons the next species, or you could have plus interactions between species, etc. So a lot of these interactions tend to be also mediated by the chemical environment. And so slowly we start to separate a little bit the way we're thinking about microbial ecology and the models we use from those of macrobial ecology. And we'll see that a little bit in this talk. So the way we'd like to go about things with this talk is that first, I will start with explaining a little bit our experimental system and how we go about measuring interactions, or how we did this so far. And then afterwards, Aurore and Ollie will both tell you more of the later work, so the more recent work that they've been thinking about on how to measure interactions in a more specific way, and also how they find, both of them, that interactions change depending on context and depending on time. And then I'll wrap up at the end. So the model system that I showed you a little picture of before is the following. So we use a sort of semi-synthetic ecosystem, if you like, because it's sort of a natural environment, but where we can grow things in the lab. So this here is a picture of a machine oil. So these are metal working fluids that you would find in many industrial settings, and these tend to pollute the environment once they're no longer being used. And so some collaborators of ours in the past, they isolated these 4 bacterial species and found that if you put them together into this pollutant, into the waste metal working fluid, that they can actually clean it up and reduce its pollutant effect. So this was what this looks like in the lab. So in the left is what happens if you don't have bacteria. So it's an oil and water emulsion. That's why it looks so milky. And then if you put the bacteria in there, they can reduce the pollution load. And so this was a nice model system for us, because we can sort of measure the function of the community, which is something that we're interested in, but that I won't go into specifically today. So what we've done then is to take these 4 species, modify them genetically so that we can distinguish them. We're able to count and follow the population sizes of each one. And then we know quite a lot about their genomes at the moment. And we've also looked at their interactions, which is what I'll show you today. So what we want then in the end is to have sort of this network of interactions. That's what we're aiming for. And remember that the way we define an interaction is the effect of one species on the growth and survival of another. And so we started together with Philippe Picardi to try and get at this problem and to measure this network. So what Philippe did was to set up simple experiments where he would take a flask, put some metal working fluid inside, and then either put each species by itself into each of the flasks, or do pairwise, all possible pairwise combinations of species. And then he followed the population sizes, so that's what you see on the y-axis, over 12 days. And here we're plotting what the species looks like if it's growing alone, so the trajectory in green, so in the color, and then in black you see how that same species is growing if another species is present. So for example, if CT is present, then it tends to sort of grow quite well after a while. And the idea then is to see if we can compare the population sizes over time when a species is alone versus when it's with a partner, and this will give us the arrow, the sign of the arrow in our network. And it's important here to note that we're using so-called batch cultures, and this will get important in the end, but just so that you know, so a batch culture is a closed flask where you start out with lots of nutrients at the beginning, and then by the end of, say, the 12 days, the bacteria have depleted everything that's in the environment. So we go sort of from a feast towards a famine, if that's a concept that's familiar to you. Okay, so how do we go about quantifying these differences in population sizes? So we thought of a few possibilities at the beginning. Our first intuition was to try and fit a mathematical model to the data. And there we had two different candidates. So both of these are quite used in theoretical ecology. So the first was a Lotka-Volterra model, and I'll introduce it in the next slide, or a consumer resource model. And the second option was just to do a statistical comparison. So the Lotka-Volterra is essentially a description of the direct effect of one species on the other. And so you can describe it with this equation here that's looking at the per capita growth rate. And this it depends on like a baseline growth rate. And then this term here, which describes the effect of species B, species beta on species alpha. And that one is represented by this parameter A. So basically we would end up with a little matrix of these parameters A that describe the effect of each species on the other and also each species on itself. The alternative is to use a consumer resource model. And there, in these types of models, we don't look at how species alpha and beta affect each other directly, but all their interactions will be mediated through the resources in the environment. So basically, if they take up certain resources or they both compete for the same resource, then that will be how they will affect each other's population sizes. And in a consumer resource model, what we do is we have one equation that describes the species abundances themselves. And here we can summarize that effect as a sensitivity function. So how sensitive is a given species to the resources that are in the environment. And then we have a second equation that describes the dynamic of the resources themselves. And we can call this the impact function. So what is the impact of a species on the resource? So for example, if it takes it up, then it would be a depletion. So that the rate of change would be negative. So these were the two options that we started looking at. And at the beginning, so we actually had quite a lot of trouble fitting the parameters of these models to our data, and none of them seemed to work. So the Lotka-Volterra failed partially because we had these decreasing functions, or sometimes like a drop and then growth later on, which didn't seem to be able to be captured by these equations. And then later we turned to a consumer resource model. And there we also had quite a lot of trouble because we actually couldn't quantify the resources in our environment since we don't know the chemical composition of our machine oil. So this was quite difficult because we don't know, for example, how many resources are available. And so we opted for a statistical comparison instead. So what we decided to do was to measure the area under the growth curve. So here for the green line, for example, we would compare the area under this curve to the black one and see if that one is significantly greater, then we would just say there would be a positive interaction, and the strength of that interaction would be the ratio of those two areas under the curves. And of course, this has its own problems that come with it, but for now, this was the best that we could do, so we stuck to this. And once we had measured the whole network of interactions, this is the result that we came to. So here, for example, you see on the previous slides with the data, I had shown you that AT grows better when the species CT is there. So that's what where this green arrow comes from. And then we found that overall, there were only positive interactions that we were able to measure. So this was quite surprising. I think we had expected sort of more of a mixture of different types of interactions. And going into the literature and trying to figure out why, Philippe had the intuition that it might be something that's specific to the environment of the machine oil, because it tends to be quite a toxic environment. And so we decided anyway, despite our difficulties of fitting, to go back to a consumer resource model. But instead of quantifying exactly the resource abundances, we thought, let's do something general and instead think of a model where we not only have foods in the environment, so a resource, but also a toxin, and see whether adding this into the model can give us a general intuition of what toxic environments could do to the interactions between species. And so rather than giving you the model, so this was the model that was developed by Björn Westman, who's a co-author on this paper. So rather than showing you the equations, I decided to show you this animation that Philippe actually made to explain Björn's model that I quite like. So here's the idea of what the model taught us. So if you just have nutrients, a species can grow. And if you put a second species in that consumes the same nutrients, the first species will grow less. Now, if you add a toxin into the same environment that kills our focal species, but the other species is able to degrade this toxin, perhaps to its own benefit, then what will happen is that this second species, our focal species, will be able to grow. And if we compare in that environment the monoculture to the co-culture, we will measure a positive interaction, because the presence of the green one was able to rescue the red species, despite the fact that they're actually competing for nutrients. And so what this suggests is that the balance between the amount of nutrients and the amount of toxins might affect the sign of the interaction that we look at. And so what Björn did with his model is that he then looked at lots of different nutrient concentrations and different toxin concentrations. And here what we're looking at is the comparison of co-culture to monoculture. So if they go better in the presence of the other species, then we see green. And then we see that whenever there are lots of toxins and few nutrients, and then when we have brown here, so lots of nutrients and few toxins, it shows that there we get more competition. So the presence of the other species has a negative effect on our focal species. Now, just to test whether this holds in experiments, Philippe went back to the lab and he basically said, okay, probably the machine oil where we saw lots of positive interactions might lie somewhere here. So if we add amino acids to our medium, this might make the environment a little bit easier to grow in because there are more nutrients, or we could remove the machine oil completely and then just have amino acids. And indeed, so we went from looking at these positive interactions in the first type of environment to more negative interactions once we added the amino acids. And then finally, in the environment that was the easiest to grow in, so the one without any toxins, we could see quite a few negative interactions. So basically the conclusion from the study was that if you have harsh environments like our machine oil, then interactions are more likely to be positive. So this was quite nice, but unfortunately the system, like I said, it didn't allow us to parameterize our model. So it's interesting to explain what's going on, but it doesn't really help us to make predictions. And ultimately that's where we would like to go, is to see what happens if we systematically change the environment, or if we want to carry this system on into the future and see what would happen in terms of coexistence, for example. And so we need a simpler chemical environment to be able to do that and really fit our consumer resource model like we wanted to. So this was the project that was done by Rita, who was doing the experimental side, and Aurore, who did the model. And so then next I'm going to hand over to Aurore, who will explain more about this part of the project.
Okay, thanks. So I'm going to share, if you can stop maybe the...
Yeah.
Thanks. Okay. How does it work? To remove this. Okay. Okay. So thanks for Sarah for introducing. So yeah, this is going to be some work I've done during my postdoc in Sarah's lab. So I'm not in Sarah's lab anymore, but we are still collaborating a bit. So mainly a project that I did with Rita Di Martino, who was a PhD student at the time. So now the idea is to focus on a much simpler system than the metalworking fluid, in which there is a lot of different compounds, and focus one specific compound that Rita has characterized, in which you have a kind of a dose dependency of the behavior of the species. So it is the linoleic acid, and at a low concentration, two of our focal species, AT and CT, are able to survive on linoleic acid and eat it. So it's a nutrient for both. But at a higher concentration, we observe that AT dies. So linoleic acid is a toxic compound for AT, but not for CT, which is what's interesting here. It's still a nutrient for CT. So yeah, so some experimental data here. Rita was able to kind of characterize this change of interaction type that was also the same as Philippe Bjorn's study. So at a low concentration, here if you show the monoculture and co-culture growth curves, you see that there is competition at a low linoleic acid concentration, while you have a rescue at a higher concentration of AT. AT alone dies, but the presence of CT allows the coexistence of the two species and the rescue for AT. So this is really this idea that you have a competition here in a less toxic environment and facilitation at a high toxic environment, so that the compound concentration can really strongly affect the interactions. So this is already something that we observe from the AUC statistical comparison, or simply visually. But what I did here was to try to find a model that could recapitulate this change of interaction and maybe generate some predictions in, for instance, what happens in between these two concentrations or in the long run. So yeah, I'm going to introduce a bit like different options for modeling that I considered. So the easier one, sorry, it's my mouse, that's a bit weird. Yeah, but what types of models can help us characterize the interactions? So the easiest one that we can consider, simplest one, is Lotka-Volterra. So here, yeah, to model, to fit this kind of data with Lotka-Volterra model, you need to really consider it as two separate environments. And so each condition separately will lead to a different Lotka-Volterra output. So it works. You can here show, so you have the data here and on top the fit. You can characterize this type of growth dynamics with the Lotka-Volterra equation, even though here it's a bit weird, the fact that you have first death for AT in co-culture, but then you can still at least recapitulate the fact that AT is dying alone versus surviving in co-culture. So you have a kind of way to quantify here the interactions, but I'm going to detail a bit, yeah, the parameters here. So first, this is the output of the modeling fitting process. Is it what we expect? So I would say that yes. So for instance, here at low concentration of linoleic acid, we see that we have mostly competition. So here we have negative terms with positive growth rates. And we see that the effect of AT on CT is quite low, almost zero, because we have a quite similar growth in co-culture or monoculture for CT. And also, interestingly, at a higher concentration of linoleic acid, you get this positive mutualistic effect with positive terms here, and also the fact that it is an obligate mutualism for AT, since its intrinsic growth rate is negative in the absence of CT. But this said, yeah, I think it was not the best model to use to have any predictions here, because I'm not sure we really learn anything with this more than simply the comparison of AUC in monoculture versus co-culture. And we didn't have any way to know if these quantitative estimations were accurate, whether This does not allow us to have any predictive power between the two situations or in the long run. So I'm going to explain a bit why I think Lotka Volterra is a bit of a problem to use in batch cultures, and that why we switched to a consumer resource model. So a simple illustration here. So Lotka Volterra is really equations that describe a kind of a dynamic equilibrium at carrying capacity, while here we have batch culture. So if you take the same parameters, but you change the initial, for instance, abundance or population size, you would go back to the same equilibrium, which is quite unrealistic for batch culture. So for instance, if you start 1,000 times higher for the population size, you have negative growth and you go back to the predicted Lotka-Volterra carrying capacity, which is of course not what you would expect in batch culture. If you start from a higher abundance, you will simply reach a higher final abundance. So it's not because the growth curves look like a logistic that necessarily the logistic model is the most accurate here. So this is why we move to consumer resource models. So here, really environmental explicit modeling where all the interactions emerge from the interaction with the environment. So what's nice here is that we use the monoculture only growth data of both species in all the different linoleic acid conditions that we have. I have to say that even though we didn't have any data on the linoleic acid concentration over time, we just have the initial time point, but it's enough to characterize these parameters. So I'm going to detail just quickly the model. So we have a classic mono growth here, with the maximum growth rate, half saturation constant, yield, and we have slightly modified it to incorporate this idea of toxicity of the linoleic acid for AT. So here we have a kind of constant linear toxicity term plus some toxicity that accumulates over time. So it's simply to capture the fact that AT first grows and then dies over the dynamics. So it's really implicit the way that we implement this toxicity. But we can have, so just like to show how the curves, the classic mono curve is modified with this toxicity. And we do so the fitting on the monoculture data only, which is not looking perfect, but at least you capture that the higher you increase the concentration of linoleic acid for AT, the less well it grows, and it's the opposite for CT. The higher you start with, the higher abundance you get at the end. So for CT, it's really a nutrient. But now what's nice with this consumer resource modeling framework is that we can really have some predictions and compare what the model predicts in terms of co-cultures to the actual co-culture data. So that's what we're going to do now. And we see that at 0.1% of linoleic acid, the prediction is quite okay, I mean, qualitatively. So you get this competition sign. So here it's only the co-culture data, but you do get the competition. It's not quantitatively perfect. And at a higher concentration, you do get the facilitation effect. So in monoculture, AT dies. Here, at least, AT is not dying. But you have a big gap still between the expected AT co-culture growth versus its actual growth in data. So here, this first model underestimates the effect of facilitation. So then Rita had this nice finding to understand more the mechanism of why linoleic acid is toxic to AT. So she found this protocol to measure the amount of reactive oxygen species, so the rate of the level of oxidative stress in the media with a colorimetric assay. And basically what you can show is that in both the cell-free and the AT-only conditions, you have an accumulation of this reactive oxygen species and not in the CT or the co-culture conditions. So we assume that CT is in a way able to cope with the oxidative stress, removing it from the environment. And that's probably the way facilitation occurs between AT and CT. So with this, we could now measure a bit more precisely the environment with this Ross colorimetric assay. So I could update my model. Yeah, just a small graph to show the two interactions with competition for nutrient and facilitation with the toxic part. So the new model equations, including for the Ross. So still we have our bacterial abundance here. So now the toxicity is completely decoupled between linoleic acid and Ross. So linoleic acid is only a nutrient for the species, and the toxicity part comes from the Ross abundance. So we assumed here for simplicity a linear term. So basically the death rate is simply proportional to the amount of Ross in the environment, but it could be saturating or I don't know, convex as well, maybe. It's just to simplify the system. And for the Ross dynamics, so the Ross productions comes from the oxidation of linoleic acid. The media, plus some and an active detoxifying part here, which is dependent on the bacteria concentration, given a certain detoxifying rate. So this would be higher, of course, for CT than AT, which is not able to cope with the oxidative stress. So what is nice now is that we can first estimate these parameters because we have the Ross only data without the cells to show how the oxidative stress accumulates in the media. And then we can use the monoculture data to feed the rest, all the other parameters here, detoxifying, toxicity, and the mono growth parameters. So now the fitting of the monoculture again is actually a bit better. So here we only did it for two conditions. And more interestingly, the predictions for the co-culture are now much better than before. So if you compare here the data with the prediction from the model, so this is not a fit, this is really the prediction from the monoculture of what we expect the interaction to be. It's much better, it's still not perfect, but at least quantitatively is much better than before. We really have this nice rescue of AT as well in the model as predicted by the model compared to the data. So as a summary, we have really characterized a bit as in the previous study, but in a much more mechanistic way because we have this simpler environment, how the balance between competition and facilitation is modified depending on the compound concentration. So first at a low, linoleic acid concentration, you have high competition. You still, of course, would get a bit of facilitation, but the outcome, the net balance of the two is competition. So that's why you observe competition. And the other way around, when linoleic acid concentration is high, you of course get a bit of a competition, but the effect of facilitation is higher. So the outcome, the net interaction would be positive facilitation. Yeah, just to check this, we added Rita added some antioxidant in the media. And in that case, this rescues AT even in the absence of CT, and AT which is higher than CTs compared to with CT. So if you remove the facilitation part, you get only the competition, basically. So just as a proof, and we also can show that in the model if you simply put a Ross amount to 0. So this is all for my part, and I will now hand over to Sara and then to Oli.
Full screen again.
Oh, sorry.
That was my fault.
Yep. Okay. So, which one's that?
Wait.
Let's
Close it again. Let's close it.
Sorry. Okay. Share screen. There we go. That one.
No, the one. Yeah, that one.
All right, thanks for that. So yeah, so Aurore's just given us a great example of how interactions can be context dependent. So depending on where you are in this kind of chemical space,
we're going to see that there are different interactions arising. So if you have high linoleic acid concentrations versus low linoleic acid concentrations. What I'm going to be talking about is how that context dependency in batch culture situations can lead to time dependency of the interactions that you measure. So to motivate this, I'm going to start with the model that you've seen already. So this is Bjorn's model that Sarah discussed. So here we have two species that we're interacting via this nutrient and this toxin. And it turns out that you can reinterpret this model as just being a single species model. So now what we have is a single species, which is interacting with other members of itself via this competition for a single nutrient and via this detoxification of the toxin. And the question that we're going to be asking is, what is the interaction of this green species with itself? So in the kind of general Lock-Volterra framework, this is kind of asking what are the diagonal terms in the interaction matrix. So that's the analogy, but we're going to see that the actual picture in the consumer resource case is a bit different. So you might think that we've already gotten an answer to this. So we've already seen this plot here. So this is taken from Bjorn's model. And this is a prediction of what you would measure as interactions at different nutrient and toxin concentrations. So this gives you this kind of context-dependent picture of the interactions that you measure in this system. And so if you're down here where you have high nutrient concentrations and low toxin concentrations, you have domination of this nutrient competition. And over here you have dominance of the detoxification positive interaction effect. But in fact, this actually glosses over quite a lot of detail. So the problem that we have here is that in batch culture systems, we have a situation where we start with a particular distribution of our nutrients and our toxins, but then over time these change, as Sarah was mentioning earlier. And so really what we have in this kind of resource landscape is an initial position in this space, and I'm going to represent the toxin concentration here as Q and the nutrient concentration as N. You start off at a particular location, and then you have some trajectory in this space, which I'm going to represent with this bold R as a function of time. So how does this, in fact, that you have this changing environment, how does that mesh with our idea of interactions in batch culture systems? So to address this, I'm going to go back to a more general case. So we're going to go back to our general consumer resource models. That again, Sarah introduced earlier. So we have these separate equations that represent the change in the resource abundances and the change in the population sizes over time. And since we're in a batch culture condition, this term, which represents the sources and sinks, so the external sources and sinks of the resources, that just goes to zero. So this disappears. So we can take these two equations now, and we can combine them. And we get this expression down here. And what's nice about this expression is it looks very similar to actually the generalized Lock-Volterra expression. So we have here a per capita growth rate of our different species. We also have a basal growth rate, which is constant. And in the case of the consumer resource system, this is just the growth rate at that initial resource concentration spot. And then we also have this interaction term, which in this case is mediated by the product of some interaction term with the species abundances over time. And then we sum over all of the species in the system. But there are two important differences here compared to what we have in the generalized Lock-Volterra case. So here, first, we have this interaction term being time dependent. So depending on where you are in this resource space, that tells you end up with a different interaction. And then we also have this time integral. So we have to integrate this expression over the entire history of the experiment up to the time t. So we start off with zero interaction, and then later on this term accumulates. And the reason for that is that the resource distributions don't change instantaneously. So you have to wait for these resources to actually accumulate changes over time. Now this interaction term is the product of these two functions that Sarah mentioned earlier, so the impact and sensitivity functions, and we call this the instantaneous interaction. So this tells you what the interaction is at a particular moment in time. And I'll explain this framework in a bit more detail using the nutrient toxin system that we were talking about before as a kind of example. So in this case, again, we have the nutrient concentrations and our toxin concentrations, and we can represent our impact function as a vector field over this space. So these arrows represent the direction in which species alpha is pulling the environment at a particular point in time. So you start off, for example, up here, and then the environment is being pulled down towards the origin as the nutrient and toxin concentrations are decreased. We also have this plot here, which represents the sensitivity function. So this is just the per capita growth rate, again, as a function of our nutrient and toxin concentrations. And then we can represent this term here, which is the gradient of the sensitivity function. And this is another vector field. So what this represents is at a particular location in this resource space, what is the direction that alpha wants the resource space to be pulled in order to increase its growth rate. So now when you take the dot product of these two vector fields, what you end up having is you ask the question, are these two vector fields pointing in the same direction or are they pointing in opposite directions? If they're pointing in the same direction, it means that the resource space is being pulled by the impact function in a direction that increases the growth rate of alpha, and vice versa if they're pointing in the opposite directions. So when we take this dot product between these two vector fields, this is what we get. So this encodes the kind of context dependency of this instantaneous interaction in this resource space. But we've already seen something that looks very similar to this. So what we've seen already is this measured interaction plot for this model. So why is it that these two things are actually different? So what are these two things representing that's different between these two? So to address that, we're going to look again at our instantaneous interaction plot and remember that we have trajectories in this space. So although we're starting at a particular location in this resource space, over time we're navigating to another point. And so if we now take, if we plot this instantaneous interaction over time, which we can do with this yellow axis over here, we see that we start off in a location where the interaction is positive, but then we move to a location in the resource space where the interaction becomes negative. And then we can also evaluate this integral. So this is the integral that tells us what is the impact of alpha on its own growth rate as a function of time. We can evaluate this integral and we can see how this relates to this instantaneous interaction. And so you see that, as I mentioned before, it starts off at 0. And then when we have positive instantaneous interactions, it increases. And then when we have negative instantaneous interactions, it decreases again. And so what you would measure from this system is over here, you would have positive interactions, and over here, you would have negative interactions. So you have this switch in the sign of the prediction of the interaction that you detect. And basically what's happening is something similar to what we were seeing before. So in this first part of the experiment, we have this detoxification effect dominating. So that's the positive interaction. And in this later part, we have this resource competition dominating. So that's giving us this negative interaction. And so what that ends up meaning is that if we measure these interactions at different time points in our experiment, so if we start off with an early time point and measure the interactions, we see mostly positive effects. But then as time progresses, we see more and more of these negative measured interactions coming into play. So this is basically because initially you have lots of toxins, but then those get degraded, and then it is the resource competition that ends up dominating the overall measurement. All right, so this is all very well theoretically, but what is it that we actually see in experiments? So as an experimental analog of this single species toxic nutrient system, we used a beta-lactam-based system as a model system. So beta-lactams are antibiotics that are degraded by enzymes called beta-lactamases, which are secreted by specific species into the extracellular environment, and then these break down these antibiotics, which allows them to grow. So this results in something called the inoculum effect, which is that the higher your initial density of cells, the higher the measured MIC of the beta-lactam antibiotic. So you can see that here in these plots over here. It's an effect that's been obviously known about for a long time, because this is from 1946, so quite well established. And so this is kind of suggestive of a positive interaction. So the higher your initial population size, the better you're able to grow in this case. And so we use this in combination with competition over a single carbon source as our model system. And this species that we're going to use is one of the members of the 4 species community that Sarah mentioned earlier. So this is C. testosterone. Okay, so we take an exponential phase culture of these cells, and we inoculate that exponential phase culture into an array of different environmental conditions. And this represents that resource space that we were looking at earlier. So we have different toxin concentrations, so these different ampicillin concentrations on the y-axis, and we have different proline concentrations down here on the x-axis. And then we also have in each of these environmental conditions, one culture that started at a high OD and another culture that started at a low OD. And the reason for doing this is related to how you measure interactions normally. So typically in an interaction measurement, such as the one that Sarah mentioned earlier, you would have two different culture tubes. So the first one would be a monoculture tube, where you have a particular population size of a test species. And in the second tube, you inoculate the same size of that initial species. And then on top of that, you add an additional population of another species. And then you ask, is that the test population doing better in the presence of that second species or worse compared to the monoculture reference? So this is exactly the same idea, except that now what we have is this second species is more members of the same population, is the idea in the high OD start case. So we can kind of regard this tube as the monoculture, and then this starting population in the monoculture tube is replicated over here as a subpopulation of the total. And then there's this gray population, which is the additional members of that same species that's added on top of that. And we're going to ask, does this pink population do better or worse in the presence of the other members of the same species? So to do this in practice, what we do is we measure the OD of our different systems over time. And you see that in the high OD case and the low OD case, you get these two different growth curves. And then you normalize the high OD culture according to that initial ratio between the inoculation densities. And so the idea here is that this represents that pink subpopulation that I was showing you in the previous slide. So now when we have this gray line being higher than this black line, that indicates that this subpopulation is doing better than it would do in the monoculture, where there isn't that additional population. And when it's lower than that black line, it's doing worse. So we detect this as a positive interaction, and we detect this as a negative interaction. And then we have this crossover point here where you can see that we go from a positive to a negative. And so to quantify this more robustly, what we can do is we can just take the ratio between the gray line and the black line. And so whenever this ratio crosses 1, we go from a positive interaction to a negative self-interaction. So what do we see in our experiments? Well, so if we now start with a kind of control condition where we don't have any toxin in the environment, we see that the low OD case and the high OD case look very similar, but they just have this time shift resulting from the different inoculation densities. And that time shift remains consistent no matter what the protein concentration is that you have in the system. And as a result, you end up with these negative interactions being measured predominantly. But now when we have ampicillin in the system, this time shift changes. And so you see that when we have high ampicillin concentrations, we actually have a longer time delay for this low OD starting culture to start growing. And that ends up resulting in what you would measure as a positive interaction. So you see that there's this dose-dependent response in this plot here. So the higher your inoculation ampicillin concentration, the stronger the positive interaction and the longer it seems to last. And then eventually it switches down to this negative interaction mediated by the competition for the protein. And we can summarize this by measuring the peak and the final interaction values. And we see that consistently over all these different environmental conditions, we do see this switch in the measured interaction. So to kind of summarize what I've just told you, it's not only sufficient to know what interaction you're measuring, so the particular combination of species you're interested in, it's not only sufficient to know what environment you're in, you also need to think about what time you're measuring your interaction, because that will also tell you something about the system. So with that, I'll pass back over to Sarah.
Okay, so back to our outline then. I'll just take a couple of minutes to wrap up. So I showed you this slide at the beginning where we were asking, sort of zooming back out sort of to ecosystem dynamics and whether we're able to predict them. And we were sort of talking about that we don't know a lot about how microbes interact, and that in general in the field, this seems to be that different people tend to measure either positive or negative interactions. And we hope that we've shown you that the reason for this confusion might be that the environment in which you're measuring interactions can really change the balance between positive and negative interactions. So what you've hopefully seen is that in all of the examples that I've shown you, both positive and negative interactions are occurring at the same time because of the presence of nutrients and toxins. And we think that these are sort of layered on top of each other. And often what you're measuring is sort of the net effect of all of these things that are happening either over time. So you're basically integrating over a certain amount of time, or also because of the relative abundance of the toxins versus the nutrients in the initial composition of the environment. And then, of course, looking a bit towards the future. So what we didn't talk about today is whether we can use this type of information and also the models that we're building and that we're parameterizing in order to make some predictions about how different species would coexist. And this is what we're working on next, both in Aurore's work. There's a part about coexistence and maybe also in Holly's future work. Okay, so then maybe just a final word about the mathematical frameworks, because we looked at both the Slatko-Volterra framework versus the consumer resource models. And just as a reminder, so in our work, we might have some particularities. One is that we're specifically looking at systems where we have both nutrients and toxins. And this is really why we see this layering of different interactions. So one question is whether this is generally relevant. And I think this is the case, because even though we might think of toxins very much from an anthropomorphic perspective, I think in any given environment, you will have things that microbes like to grow on and things that they don't like so much. And so I think this combination means that there will in practice often be these types of nutrients and toxins. So if you think of a gut environment, there might be antimicrobials that are being produced by a host that will kill off certain species, and maybe others can remove these antimicrobials, etc. Then we should also mention that so far we've been working in batch cultures. And so a lot of the implications that we see are specific to this batch culture environment. And now we're also moving towards doing continuous cultures, where you can basically fix yourself in this space of nutrients and toxins and try to keep the environment constant in one place, which might actually help to understand and measure interactions. And then finally, we've also been moving towards simplifying our chemical environments to help us to predict things using the consumer resource model. So again, just on sort of which model should one use in the future, let's say it's very dependent. In a lot of our work, we found that the consumer resource model has been the most powerful, but Slatko-Volterra is still often a really good tool because there are few parameters, so you don't have to understand exactly the mechanistic side of what's going on in your system. So it's relatively easy to estimate and measure. But what we've learned is that it might not be suitable for batch cultures because the interactions will be changing over time, and also because of a lot of the points that Aurore was making in her part. And we also find that it's quite difficult to capture things where the growth is basically changing over time. So where you might have growth at the beginning and then death later on as the toxins accumulate. That's very hard to capture with Slatko-Volterra. On the other hand, the consumer resource model gives a more mechanistic understanding of what's going on and what's mediating the interactions. And in that sense, it has greater predictive power. And one can also sort of add chemical blocks, if you like, as one understands, as Aurore showed with the reactive oxygen species. But it also has more parameters, so there are more measurements that need to be made. However, once you understand the chemistry of the system, it might scale better when you start adding species, because you just need to understand how the species interact with the chemistry and not with each other. But it still might not be suitable to complex environments like the metalworking fluid that we started with at the beginning. Okay, so just to zoom out then all the way back to the natural environments, we think that by sort of combining experiments and hopefully the appropriate modeling framework, depending on the experiments, we should be able to get to a point where we're able to predict the ecological dynamics of microbial communities, such as the ones on the slide. So finally, I'd just like to thank quite a few people. So people from our department, from my postdoc, and also collaborators. And then, of course, very importantly, the current members of the lab and also the former members. So Aurore is here on this list that have contributed to all of our work, and especially the funding bodies and also the taxpayers behind them. So thank you. I hope we haven't gone out over time too much, and we'll be very happy to take your questions.
Thank you. Perfect timing and great talk. So maybe now I will stop also recording.